a brown fringe at the edge of that white sheet, and it sits kind of like you'd see a shelf mushroom. You know those that grow on trees? Yeah, that did that on the side of rocks.
I think we saw some of those on the Cordell Bank and Fowler-Own Dives.
Oh, neat.
Something similar.
I would believe that. That's similar kind of habitat. I was watching some of those dives.
And it was a glass sponge?
Yeah, now I want to smell that sponge. That sounds amazing.
Yeah, I wish I
notice
would
you
have
guys-
smelled them now.
Well, you should probably smell lots of things. I mean, I smell sponges because that's what I study. And sponges produce a lot of secondary metabolites, which are compounds that are produced for
We've got an octopus coming up.
we have an octopus.
Top center.
Yes.
He's got his arms displayed out for us.
Yep.
We also see a grenadier, that fish swimming in front of us.
What are all the dark strands? Are they worm tubes with shadows?
No, there are things projecting up off the, like small corals.
So it looks like the third arm of that octopus is hidden. And by looking at the tip of the arm, we can tell whether it's male or female, but we might not get a chance to see this one.
It's just sort of lightly touching the sediment.
All right, thanks for zoom. Ooh.
Oh, wait, we're going to see it. I think I might already see a little bit of a... Okay, don't get antsy.
All right. If we have to go, that's fine.
Yeah, there was no zoom. It was just good pilot driving.
That was excellent pilot driving, Dan. I think I see the pseudophallus.
Would you like a zoom?
I don't know if we can see it.
Let's see if I can get.
We're on the wrong side of the octopus.
Yeah.
But that's okay.
Yeah. All right, coming wide.
Thank you.
Okay, full wide.
Very cool.
I can try and come around.
Nah, that's okay.
It's okay, we can keep moving.
Yeah.
The current's behind us, so we have a tailwind. We're trying to get the boat up to ludicrous speed.
Yeah.
Ludicrous
Did you just say
speed sounds like a
ludicrous
good-
speed?
Sure did.
I know that reference.
Amazing.
Oh, tin
That's a tin
can.
can.
I'll let you know when we've gone to plaid.
All right.
We'll see it on the screen, right?
Full wide.
Yeah, that's how it works. Oh god, I'm ready. Yes, all of us are Googling sponges right now. What kind of sharks, if any, can live at these depths? So we've got a number of deep sea sharks, 6 gill, 7 gill.
There's a
No,
sea pig.
be cool to see a gulper shark. I'm not sure if
Octopus.
they're
Where? Oh
That
yeah,
one's
octopus on the right.
dry, actually. They might be deeper, but that would be super cool.
Oh, we'll be able to ID this one.
Excellent.
Yes.
That's right.
True.
Premature zoom.
Well,
Sorry.
looks like a
Coming
male.
wide.
Oh,
Well,
that's
that-
the second limb.
The third
Full
one is
wide.
curled under, isn't it?
It
Yeah,
is,
it's
but you can
protected.
just see the,
Okay.
it is
push
behind there, but I think that little light colored tip on it is,
Oh, yeah. It might be.
Heterocotyledus?
Hectocotylus.
Coming wide.
Hectocotylus?
Yes.
Could we,
Full wide.
Thank you.
type?
I think it's time I look up the etymology of that word.
Yeah.
What is it? It was false. Not false arm. False.
Oh. This says hecto was 100 and kotul was hollow thing. So it's 100 hollow thing.
That sounds weird, but okay.
Or
That's-
small cup, not hollow thing. That sounds better.
Yeah,
'cause
small
it has
cup.
A cup-shaped pseudophallus at the tip.
Okay, so I have a lot of people requesting some more information about sort of the geological structure we're looking at. How did these cliffs come to be?
We've reached
What
0.5.
sort of
We wanna go faster
texture
than
and
0.5.
clay and rock are we looking at?
So we are at the outskirt of Davidson Seamount, which is a extinct volcano. It's no longer erupting. It last erupted about 9.8 million years ago. And we know that because the geologists have aged the rocks along the seamount. Now what we're on now is probably a thin layer of sediment and overlaying maybe a thicker or clay-like sediment. We haven't done a lot of push cores out here, but the ones we've gotten, that's what it looks like. So right now we're just looking at a bunch of sediment.
The cliffs look like they have been the result of slumping caused by gravity operating on.
Of the clay sediment.
Yeah.
Yeah.
Mm-hmm.
Yeah, some of the cliffs that we saw earlier didn't have loose dirt on them. They looked very smooth, like they'd been sheared.
Right. Here's some stuff coming up.
All right, let's see what we got.
Yeah. Well.
I do wonder how mounds form.
Yes.
Makes those.
Right.
I imagine animals make some of them, burrowing animals.
Oh yeah.
But you mean like the big mound we're headed for?
Or
Or
that
little?
too, but at this moment I was thinking these small ones.
Yeah. Do we call that bioturbation? Yeah, that looks kind of lumpy, doesn't it? Somebody
Yes.
got in there and worked around.
Swimming through the snowfall. This is marine snow that you guys are seeing here. So it's a mixture of all kinds of things from the surface. So dead plankton, fecal pellets from plankton, aggregated phytoplankton cells that have sunk down. There could be some whale poop in here. All kinds of stuff just kind of sinking down as particulates that are the source of carbon and food for the animals that live in the deep sea. And it's estimated that every 1,000 meters or so, there's about 10% of the food that was 1,000 meters above it. So there's not a lot of food left by the time we get down here. Our current depth, I'm going to load up that screen, is 3,300 meters.
Perfect. I was just going to talk about that. Very nice. What is the goal if you've already mapped this area? We kind of have mapped this area. We were going to continue to map this area, but we need to explore the habitat and the biology and the geology and the chemistry, and we want to know all the details about what's going on down here at Davidson Seamount.
When we talk about mapping, we're talking about the shape of the seafloor and the depth of it, not what lives on it.
So that form of mapping was done using multi-beam bathymetry, which is sonar that shoots different beams of sound out in kind of a fan, and it listens for the bounces back of the sound against the seafloor. And so bumps would reflect the sound back sooner than flat spaces, and it calculates, therefore, the shape of the seafloor. But as you can imagine, the sound needs to bounce off of something that's a hard structure, like rock or sediment, not a soft-bodied thing like a coral or like a,
Octopus.
octopus brooding ground. Yeah. So it's important to know what the seafloor looks like because that gives us hints of what regions might be similar to each other based on how they look on the map. But to find out what habitats actually exist there, we have to be able to see them.
And then we can use that information to predict what organisms might be living on a map we haven't explored with an ROV yet. So it's sort of circular.
Mm-hmm.
Ooh, that was a great way to explain it, you guys.
And some of the multi-beam is so high resolution now that we can actually start to see corals in the mapping. It's quite amazing.
Oh,
You'd
I
need
love
an
science
AUV for
circles.
that.
Yes.
We would like to know what the oxygen and temperature level is currently.
The oxygen and the which?
Temperature.
Oxygen temperature. I
Ah.
think a lot of these things should be Is the temperature on Nautilus Live, or the oxygen on Nautilus Live? I know sometimes the temperature was up.
Well, I can take a look at that.
I know we have depth.
We have depth. And we got temperature. We got depth, temperature, speed. Ooh, and depth for both of our ROV
Oh, I don't believe that speed.
babies.
That speed's not right.
Oh, it's wind speed. It is not a
wi-
speed. It is
wind
a wind
speed is
speed. That makes much more sense. But we do not have oxygen.
Well, the oxygen concentration right now is 117 micromoles of oxygen per liter. And the temperature is 1.7 degrees, which has been pretty constant since our last watch.
Brr.
Celsius.
It's
Celsius.
A little chilly.
It's a little bit cold. I did some work. Well, I've done all of my research in cold water. And so the waters around Monterey Bay are about 8 degrees up to 14 or so, depending on whether there's upwelling or not. And that's cold. But then I did some work out in Norway where the water was 6 degrees. And that, if you put your arm in too far, it sends this chill through your whole body.
Oh.
I can't even imagine what 1.7 degrees would feel like.
Oh.
It's like insta-freeze.
Yes. That's
It's
too
one-
cold for me.
It's one thing to be an air degree. When it's water degree,
It's true.
Yes.
it's shocking. Right.
Shocking
Yeah,
is right.
Pulls
Yeah.
all of the heat out of your body. And yes. Very quick hypothermia. That's why lots of our whales and seals and mammals have blubber, because it helps insulate their bodies from that cold water.
And that's why humans wear thick suits filled with warm water when they go into water that cold.
For those of you who are wondering, when we encounter animals, do we sort of bother them or not? Some of them don't seem to mind. Some of them are kind of too slow to run away. Some of them are blind and may not see us. We do make some noise, so we might be scaring away larger things, but we're not really sure because we can't see them out of our range, and if we turn the lights off, then we can't see anything at all. So we're not totally sure, and we don't have a lot to compare it to, but a lot of the creatures don't seem to mind us drifting by, so.
And some of them are attracted to the ROV, whether it's the lights or the motion.
Oh, that's true.
Yep.
Like scavengers, like these grenadier fish are scavengers, and they'll come up to the ROV to scout us out.
Very cool. We were just talking about the wind speed on Nautilus Live. That is like the actual wind speed on the surface. Nothing to do with the water down here. Well, it's all a big system, but that's the wind speed that the boat is dealing with, way up. What would you do if a mermaid swam up to the camera? That's not happening.
I would believe that the Earth is flat.
That's what would happen. Yeah, there was a discussion before of we can't necessarily prove things are not there, but we have a lot of evidence to suggest that those things don't exist.
I would ask my fellow scientists in the van if they were seeing the same thing, because I wouldn't believe it.
I wouldn't believe it either. I would think Wayne had switched our video cameras somehow. Suddenly we have a feed that I, no. Okay. Everyone is asking for some more sponge facts. So,
Sponge facts.
Amanda, what is the oldest sponge that you've seen or known about? evolutionary, the ones that developed the furthest back that we know. And can Amanda tell us some of her favorite evolutionary biology facts? I want to learn.
Oh my goodness.
That's
oh, God.
great.
This
Okay.
is awesome. Thank you,
Okay,
explorers.
But I have to be correct. So let me just check the number because I do have the record of a sponge that its age was estimated using isotopes to date it. And I want to make sure that I get the right name of that, or the right age of that sponge.
So a living sponge?
It was a living sponge. Yeah, I know they also asked about the evolutionary history. We'll go back to that.
Very cool. Question, if we saw a giant or colossal squid swim up to the camera, I think we all would be incredibly excited.
Yes.
That would be such a day. We have not gotten the giant or colossal squid on camera all that often. We usually get sort of bits and pieces and a quick scoot by, but it would be fantastic to be able to see such a large squid.
We have seen Humboldt squid over Davidson Seamount during warm water conditions. And we actually have tried to fish for them above the seamount with students from Stanford University. Unfortunately, we got skunked.
Oh, they're really cool. They kind of scare me though.
They are large.
They are large and bright red, and they're fast and they're strong, and I'm not that coordinated in the water.
Okay, Amanda might have some information.
Oh,
I
let's
do.
Yes, we're ready.
So there was this very neat paper that used radiocarbon dating to estimate the age of the glass sponge Rossula rakovice, and they estimated the sponge at 440 years old.
Wow.
There have been sponges that were larger than that one, so it's possible that some sponges can be even older, but it's also possible that they just grew in a different way or at a faster rate.
See, now that's why people don't have pet sponges.
Yeah.
Because they would outlive their owners.
Oh, it's like the problem with turtles. You know,
Yeah.
my friend has a pet turtle. They had to
Parrots.
make an agreement and have a plan for
Yeah, for
the
its
turtle outliving them.
hair, its care, yes.
Yeah. Now, evolutionarily, sponges were definitely present during the Cambrian explosion, which is when many of the animal groups that we now know today were also present in the fossil record. And they likely were around in the period before that, called the Neoproterozoic. Although fossils that keep on trying to push that date back are sometimes, well, I don't believe them. I think they have to be taken with a grain of salt. So 630 million years ago, sponges came about, along with many other animal groups. And evolutionarily, the sort of bonus of getting to study sponges, I became interested in them because ecologically they're really important in their habitats. But evolutionarily, they're also one of the first animals to branch off from the last common ancestor to all animals. So learning about them and how they work tells us some really, really basic ancestral characteristics that we can have too. And so it's really fundamental stuff. Like
Just to pause, that's a giant cusk eel that we're seeing in view. Don't mean to interrupt, but I didn't want the viewers to miss out saying, what is that? What is that?
That's great.
You know we'd get the question, so perfect.
We see a lot of these down here. So, sorry, take it away, Amanda.
You're good. You're good. You know me. I'll just keep talking about sponges. It's probably good to stop me.
I have a question when you're done.
Oh, okay. Well, when we look at a sponge, I was looking at how much energy does it take for a sponge to be a sponge, and what are the sort of energetic costs that they have? Because if they're having this big effect on their environment, we want to know kind of, how much food does that take? How much energy do they spend? And that was a big focus of my research. And during my PhD, I started looking at how quickly they produce cells and then replace cells in their body. And that's something called cell turnover. And in a tropical species of sponge, it was suggested that that turnover rate is extremely fast. Something like they replace 1/3 of the cells in their body each day.
Wow.
Which just sounds astonishing. How much energy would that take? I'd have to eat
I'd be exhausted.
a lot of pounds of
right.
food.
Sounds tiring.
Yeah. So,
You'd spend all day in bed.
you'd have to spend all day eating. So,
Well, that would be fun.
Isn't that what we do on the ship?
Basically.
I'm working on
Well, we work. No, we play the rest of the time.
If
Eat and play.
only.
Eat and play.
that. Yes. So I looked at several species of temperate sponges that live in colder water, and so you might expect to be lower energy because it's colder, so they don't have as high of a metabolic rate. And I surveyed across them and found that their cell turnover rates were much slower, on the order of about a week for cells to kind of turn over, the cell lifespan, if you will. But also that cells were being replaced by not just simple divisions, but by stem cells that were coming about. And these cells I was looking at were the choanocytes, the feeding and filtering cells. Sometimes we would see a little mobile archaeocyte, different sponge cell, move up to the choanocyte chamber and then differentiate into a choanocyte and wiggle its way in. And that is a very stem cell-like thing to do. And being one of the first animal groups that branched off from our last common ancestor, it suggests that being multicellular meant having a stem cell population that would be responsible for replacing your cells. You couldn't just have cells dividing willy-nilly, because that would mean that you don't end up with a fully multicellular being that's functioning and cooperating together.
So is there one stem cell type that differentiates depending upon where it wiggles into?
That one I'm not sure. So there's a lot of work being done right now using single cell sequencing to understand the different cell types that are in sponges. Because morphologically, many of the cells that we call archaeocytes are kind of different, but we can't differentiate them well enough just by looking at them. So the ones that migrated in to form choanocytes were usually small archaeocytes, and there were also much, much bigger ones. And so we call them all archaeocytes, and they were amoeboid.
So
But
what
there might be
happens if you transplant an archaeocyte from one species into the tissues of another species?
Oh, well, sponges do have innate immunity, so
Okay.
if two species touch, they won't mix.
Interesting.
Although, if you take a single sponge and grind it up in a blender and then put it into a container, depending on the species, those cells can re-aggregate back together, and for some species, reform the canals that are necessary to pump water through it.
Wow.
But not for all.
Wow.
So I had a question, Amanda, about the fossil record. So if you're looking at fossils, what is left behind? Is it the spicules that are left in the fossil record, or the whole sponge is left in the fossil record?
It depends on how the spicule, or I'm sorry, how the fossil was made. So a lot of the time the spicules would undergo diagenesis, which is where the glass of the spicule gets replaced by other minerals. And so you end up with these sort of casts of the spicules arranged, and the soft tissue has a different form of diagenesis, so you can see the preservation of the soft and the hard. And in other ones, just the spicules are left behind.
Interesting.
You can, for those who are watching from Europe, I think it's so cool. You guys have the Tethys Sea, like uplifted into your mountains. And so if you go to the mountains in Germany or in northern Italy, you can find sponge skeleton fossils from sponge reefs that existed back in the Jurassic period.
Wow. Wow.
When dinosaurs were roaming the earth, there are sponge reefs
Wow.
in the ocean. So cool.
Danny, can you tell us what countries are tuning in right now?
Absolutely. Sorry, I was getting off on tangents reading about sponges.
I do it all the time. It's cool.
Amanda has that effect on people.
Yeah, I am so excited. I love working with people that are so excited about what they do because it just makes you excited, and then I learn so much more. And guys, I don't ever want to leave this ship.
I can numerically quantify her excitement by the number of dB that I have to drop her microphone.
Sorry,
She
Wayne.
is by far the winner of the dB attention.
Well, that
Attenuation.
because that's also my initials.
Required.
So that sounds fantastic. Okay, so the countries. We have the U.S., Canada, United Kingdom, Australia, New Zealand, Germany, Singapore, France, Brazil, Ireland, South Africa, Portugal, India, Indonesia, the Virgin Islands, Sweden, Belgium, Puerto Rico, Peru, Netherlands, Malaysia, Korea, Iceland, Guam, Greece, Spain, Chile, Bermuda, and Austria.
Wow.
Amazing. That just gave
Welcome,
me chills.
everyone.
We got the whole world looking.
We have
I
yes,
think they tuned in for the whale fall.
the whole world.
And they're sticking on for the
Yes, I think
Sponge
they
touch.
are, and we are
The
so
sediment crawl.
glad to have you guys out with us. I know I'm getting lots of questions in. Let me just do a quick update of where we are. There's a lot of information posted on the page where you guys are watching the videos about, Nautilus Live. It's got our speed, it has the wind speed. It has the depth of our 2 ROVs. It has the temperature. There should also be a map in the corner of the quad view that will tell you where we are. We are currently traveling through Monterey Bay National Marine Sanctuary. We're skirting around, I believe we're still on the southeast side of Davidson Seamount. And we are exploring an area, kind of trying to characterize it, understand a little bit more about the flora and fauna that live here, including geology, chemistry. The lasers are on. These lasers help us measure what we see, so it gives us a good size perspective of things. They are 10 centimeters apart, or about 4 inches. So that's a great way to give you some perspective on what we're looking at. I think that covers a lot of my questions. And then we can go back to sponge questions when Amanda's ready.
Is it possible to get bubble to point at the top of the camera to show the lasers?
I do have a question of how I became a science communication fellow. I applied. So
Oops.
I did study
Not now.
science. I got a bachelor's degree in marine science, and I have a
Okay.
master's degree in coastal science studying
Can't
shark
see 'em.
stress physiology. I absolutely love sharks, and that would be my goal, is to just have a shark science communication life. So if anybody out there wants to help me start that business, let me know. So I loved all of the science, but as Wayne just said here, I get really excited about teaching other people the really
Thanks.
cool things about the ocean. And so I switched from purely research scientist to education and outreach and science communication. And so I'm sort of in the middle between those two fields, and I absolutely love it. So if you are interested in doing that, or interested in any of the many positions that we have out here on the Nautilus, right on the main page, it says explore with us, I believe, or join us. Get involved, that's what it says. Get involved, explore with us. And there are applications, and you can click on that, and that will give you plenty of information about all the different positions that are out here that you could join the Nautilus. And we're always streaming when we're out diving. Unfortunately, this is our last dive of the season.
It's our final dive of the season.
Final dive of the season.
Quiet.
You're gonna upset Frank.
Don't give it a name.
When does the new season start?
Okay.
There's bad luck calling it that.
Okay. We're
It's
diving.
the final dive.
The
It's our
final.
final dive.
We're the final countdown. Does that work? Thank you. We will not be lowering the ROVs into the water with a live stream after this evening.
And Danny-
Until
Yeah, when?
next
When's the next
year.
When are you back on?
When are we back on? We don't know the exact date, but it should be sometime around May 2020. So we use the fall time to get the boat all set and ready. We actually are getting a brand new engine, so that's going to be super nice. We get all of our scientists and all of our plans and all of our people ready, and then we head out. So there's lots of time for you guys to get involved, and there is so much information on the website. You could probably watch Nautilus live videos for the next 20 years and be happy. There is also a ton of educational material if you want to learn more. And for teachers who want classroom lessons, we have a ton of stuff up there. So there's so many resources. And then there's all of us that you can ask questions from. So thank you so much for being a part of this adventure. We're so excited that you guys are exploring with us. And then we get to explore with you.
So Miles, about how far are we to the pinnacle?
To the pinnacle.
The cone?
The cone.
Yeah. To the ROVs.
1,000 meters.
By Sea Crow is 1,500 meters.
Okay.
All right.
By Sea Crow. We got to make a turn in there.
But it's going to be a fabulous turn. There might be something exciting to see there.
That's right.
Did you say sea crow? Like by the...
As the crow flies.
As the sea crow flies?
As the sea crow flies.
Got you. Very cool.
Sure, we are making our way to a cone that we mapped previously, and we're hoping that it has some high relief and maybe a wall or 2 or 3 or 4 of corals and sponges and everything in between. Right now we're just traversing this sediment plain, or I don't know if I should call it a plain, valley. So we see a lot of soft-bodied organisms, including sea cucumbers and tube worms and some crabs or squat lobsters on the surface. These dark red spots, round spots, are benthic jellies called benthocodon. There's one to the right and one near the center lasers that are swimming by. And a little snailfish at the bottom center, just out of view.
He was cute. Okay, I had a question about coloration of fish. They don't have counter shading down here. There's very little light that makes it down here, so a lot of them are trying to be red, which a lot of them are red. They're not trying. A lot of them are red, and they that red wavelength does not make it all the way down here, so they become sort of a dark grayish black, purple, and then it's very hard for anything else to see them. So that's how a lot of the creatures down here are blending in. They're just having darker colorations. You know what?
An octopus on the left.
Yeah, there's an octopus on the
We
left.
could
Hi, octopus.
view into that octopus on the left. That would be great.
Shout out to STEM women. STEM women are cool. Thank you, guys.
Let me know when you're ready, Dan.
Roger.
We are so excited to have all of you joining us.
Yeah, there's a
Heather
lot
is
of
still
cool STEM women
waiting
aboard.
on a bryozoan. We're trying.
Okay, can push in there a bit.
Okay.
So we're
That's
looking for
good.
the
Hold
third
that.
arm from
Oh.
the right eye. It's curled away from us.
Oh, I bet it's a boy.
Might be protecting that third arm. They
Kinda
tend to
looks
do
like
that. Yeah?
Has a different end.
Yeah, that looks
Yep.
male.
It's a boy.
There we go.
Huh.
Jen, you're offline.
Coming what? Oh, there we go.
You're offline. That's okay, though.
It's a nice steady shot there.
Yep,
All right.
that's a guy.
Thanks for the
That's
zoom.
right.
It's missing part of
All right, coming wide.
You get those steady shots when you auger the ROV into the mud.
Yeah. How's that? Okay, I think I'm full wide, yeah.
Want to remind everybody that we have our robots diving for us, our remotely operated vehicles. We are actually on a ship above on the ocean.
Zoom on the shrimp, Mike.
Sure.
Controlling
Oh, there's
these
those
robots. We
floating
have
jellies.
some fantastic ROV pilots.
He happens to be
We've
swimming
got
against
a
the
video
current. So.
engineer. We
Fine, we
have
can
a
swim.
mapping for our
Navigation.
Thank you. Navigation. We've got some fantastic scientists. We've got data logger. We have all
Moving
of
on.
Okay.
what? We have a fantastic team working here to figure out all of this, but we are not the ones actually on the bottom. We have our robots down because we can
still on
have
the
them
ground.
down for a long period of time.
Wow,
Without.
lots of those jellies.
Yeah, there's a lot of them.
Ooh, they're
Wow.
so cute.
Wow.
Little UFOs.
Yes.
These jellies are specialists at living near the bottom. It's called
Well,
the
these are
demersal habitat.
Identified flying objects.
That's right.
IFOs.
They look like little space saucers.
They do.
GFOs, gelatinous.
GFOs,
Oh,
yes.
swimming objects. Okay, drifting. Now we're just making up adjectives to make it fit.
We got a Southern
Yeah.
Miss to the top shout out. Thank you guys for Southern Miss fans.
Hey, that's where you're from.
That is where I'm from. I now live in Mississippi. Jersey girl at heart, but I love Mississippi. Oh, we've got some good questions. I can't get through all of these. Quick one, was there shark evidence on the whale
One knot.
fall?
That's what one knot looks like.
We didn't really
Wow.
see any, but it
Like it.
had been a lot
it.
of the big blubber, lots
Giant
of
dust trail
had
behind
been, sort of devoured and digested already, so they may have been there when it was brand new.
What's that?
They kind of headed out after that. I find the ocean really interesting, but it gives me an uneasy feeling. What vibe do you get looking in this deep underwater? Ooh, that's a good one. I think Amanda's sound just answered that. It is awe and curiosity and fascination, and I feel like fear has a place in that. You fear something, and for me, that makes me want to learn more about it, and then it's not so scary. So it's just awe-inspiring is how I feel having this opportunity and being able to explore depths of the ocean that have never been seen.
And I just want to share we were doing an interaction with a bunch of classrooms earlier today, and our chief scientist, Chad, was talking about how he got interested in the ocean, and he said that it was fear, that when he was young he was afraid of the ocean, and that he felt the best way to get over that fear was to understand it. And I thought that was really inspiring. Holy cow, look at him now. He's actually been down to in a submersible, so he's actually gone down to the octopus garden depth, so 3,200 meters.
Wow, that's brave.
That is a Phaeodarian
Okay,
Ha.
thank you.
Yeah.
drifting along.
It's so clear.
It is.
Alrighty, so I have to go
And
run
as a
and
protest,
do an interaction with a bunch of other science communication people, actually previous science communication people that were on Nautilus. But while I'm gone, I think you guys will have plenty to talk about. I've got 2 questions for Amanda before I head out. Is there a multicellular sponge? And then question number 2 is, oh God, I lost it. Can Amanda explain where scaphopod tusk shells come from? So I'm going to leave you guys
where
on
do they
that
come
note.
from?
And
They're
I
scaphopod.
and I'll be back.
Okay, we'll miss you, but we'll see you soon, Danny.
Thanks. Don't find anything cool.
Too late. I'm looking at a crinoid right now. Too late. Oh wait, no, that's not a crinoid. Is that? Don't know what we're seeing.
Is that a sea pen?
Oh,
I
it's
don't know.
No, I don't think so.
Well, there we have it. Mystery. Oh, there's a sea pig down in the bottom left.
There is.
And in front of us.
Oh yes. down and to the right of the lasers, about a quarter of the way to the from the right of the screen. We had sea pig, which was a type of sea cucumber, and they have hypertrophied tube feet, so they're really, really enlarged tube feet that they use to walk on the seafloor, and that's why they look like stubby little, well, pigs with legs. So to get to the questions that Danny left behind for us, I'll tackle the sponge question. I don't know if you guys want to jump in on the scaphoid question. But for sponges, the question was, is there a multicellular sponge? And the answer to that is yes, all sponges are multicellular. So they are animals, and the way that we define animals is multicellular organisms that are heterotrophic, meaning that they don't produce their own food, and that form through the formation of tissue layers during development. So that means that they have eggs and sperm that when they get fertilized divide and divide again and again and end up making layers of cells that can be differentiated into other parts. So all animals have that functionality. They're not the only multicellular group. There are actually several times that multicellularity has arisen. One other very large group where multicellularity exists is in the plants. So plants are multicellular, but they arose separately from animals. And they're phototrophic, so they make their own food. Octopus, watch. There's octopus to the right of the lasers.
So, Jen, then obviously there can't be a unicellular sponge, but do we know what, and I can't remember the new name, xenophyophore.
Yes, xenophyophore.
No, but
But it
the
has a
name
new
has been
name now.
changed. Yes.
Monothalamians.
Wait,
Yes.
they're not xenophyophores?
No,
No.
we got that word, yeah.
They've changed. Oh,
When
welcome.
did they change? Hi.
We have a different data person.
Sorry.
Which I was
I'm
like,
just
yes,
relieving Taylor Ann.
That's
And
okay.
do we know how those guys eat?
Yes, but let me check the details. I know that they are part of the sarcodines within the protists, which use pseudopodia, which are basically little finger-like processes to come out of their shell and capture food. But what
Okay.
they actually catch, I will check.
And following up on the scaphoid, the tusk shells, the mantle tissue produces the shell. So that's where it comes
Like
from.
any gastropod or bivalve.
This is super fun. I'm like, I haven't seen any of you in the control van. Hi, friends.
Hi, Megan.
Hey, Wayne.
You require quite a bit more boost than the previous SCF.
Yeah, that's probably true.
Oh, gosh, sorry.
It's okay.
It's been quite the day. Quite the day. Got a little dinner leave happening. Danny is down to connect with the SCF's cohort interaction.
And we've just been chatting about monothalamians and scaphopods. It's been great. So I did check about monothalamian or xenophyophore feeding, and just like amoebas, they use their pseudopodia and they kind of root through the muddy sediments that are on the seafloor, and they have this sort of slimy substance that is with those pseudopodia, and when they find food, they just envelop it, and phagocytosis brings it into their bodies.
Okay.
Pronounce that word, the name of it again.
Monothalamia?
I would say monothalamia.
Monothalamia.
You know what's fun about Latin? It's a dead language, so the pronunciation is variable. So that's how I would say it, but
So monothal... No,
Yeah, you could
you
say
could
any
make
way.
up. Yeah.
When I go to the World Sponge Conference, it's always really interesting talking to people about the same species of sponge because we'll say Geodia, and my Norwegian colleagues would say Geodia, and then others might say, Geodia.
Geodia.
Yeah, so we all kind of know who we're talking about, but all are valid.
Yeah, interesting, right?
I'm sorry, was that a Zoom request?
Negative.
No.
It's been so fun having all you viewers with us all day today. Thank you for tuning in and hanging out with us on the whale fall and across the seafloor. Totally interested in your questions and incorporating them as we can here. We are on this transit back on this sedimented plain on our way to a cone on the seafloor. Looking to investigate this next really cool feature. This was exactly the kind of exploration we were doing when we encountered the whale fall just a little bit earlier. So keep your eyes peeled. You never know what we could see next. Interesting question of would you recommend pursuing a master's or PhD program for someone who wants to make a career in researching marine biology? I think that really depends. My personal take on that is it would really depend on what role you want within that. A PhD, if you want to be leading and directing a research program, is probably very required. But if you are interested in the fieldwork aspects of that or in the writing and storytelling elements, I think it's really important to consider what you want to be spending your days doing, what kind of activities you are most interested in, and then align the career to that. If you are really passionate about sharing your science with your community, that's increasingly something more that academic researchers and faculty do, but it is not sort of a day-to-day responsibility of their work. If you are really passionate about, understanding mechanisms and maybe like the I don't know, gene expression of a particular animal, how does it make its living in a certain environment, then certainly some of those entry places, some of the level of research and understanding that you might want would be at a professional science background, which would be a master's or PhD level. If you want to check out our career pages, there's a lot of detail there about the different roles on the ship and how people got them, the types of education pathways that people pursued to make it to these positions, and with some recommendations for what people would say to have a career like theirs. We have lots of folks writing in that they did not get a lot of work done today because they were here on the seafloor with us and the whale fall. I respect that. I understand that.
What do you see?
So Aaron, how far off are we from that cone feature? Oh, sorry, you're not on.
Ship or Herc?
I'm interested in both.
All right, ship to the main cone feature. The ship's about 400 meters off. Vehicles.
Zoom in there, video.
Yeah, I'd say maybe 10 minutes, 15 tops.
Straight line on the vehicles, 700 meters, but we're going to be doing a bend. So what's the status? Yeah, we're still doing a drive-by on this, right? That was the update?
Mm-hmm.
Okay.
We're just going to do a drive-by, or we'd like to see it.
Okay, come on. Try and find something better than that.
Yeah, thank you. I appreciate that. Yeah.
Wow.
You
zoom in there?
Yep, here we go.
I'm estimating about 300. Where do you get your layback information from?
So that, I think, is the ghostly grenadier.
Why
It
can't
tends to have a white head in front and then the modeling towards the back. So that is Corphonoides leptolepis.
I love Corphonoides.
This is a
That's
cool
one of my
one.
favorite.
Yeah, I haven't
Yeah,
know if
yeah.
I've seen
Colors.
one this small. This is great. Thanks for the close-up.
Okay, coming
All
wide.
right, full wide.
That's awesome.
Full wide.
Aaron, we're moving at 0.8.
Whoa.
Is that correct?
Ship is dialed in at 0.8. Right now we're achieving just over 0.5.
Roger.
You guys are 0.3-ish. If we want, right now you're about 100 meters off of what's showing up as high slope. So we might want to do an offset on the line to get you closer as we pass this feature. Unless you think otherwise, this is the situation.
I think it should be all right. I don't know what you think, Mike, but Herc can go out closer to that slope, and Argus can stay a little downslope of it.
Yeah, let's try that.
So keep it as it is.
Adjust as necessary when they get in there, yeah.
Okay. The ship is still on the move, and we'll have to adjust bearings soon to head over towards that cinder cone.
Zoom in there.
Yep, zooming.
That's a good-looking benthicodon there. Benthic jelly, and there's a sea pig on the left. I'm not sure what that small thing is. Small pinkish white.
Might be another anemone.
Ah, yeah.
Last time we saw these benthicodons, they were like more reddish in color.
Yeah, we had noticed that the
There's
distance.
a purplish red
Yeah.
hue to them.
Okay.
If they're farther away, there's less red that kind of makes it back to the lights, I think, of the vehicle.
All right, full wide.
Coming wide.
Awesome. We are seeing a lot of the pits and holes in the sediment. We don't know exactly what those are for, but likely evidence of bioturbation that means something has been digging around. There's all kinds of different worms and invertebrates that make their homes.
Straighten
in the
that
soft
camera up
sediment.
over the porch, Jess.
It's off screen.
There are a lot of fish with that elongate body form. It is part of the reason I've heard described there is these are animals without a massive amount of muscle mass that they're carrying around when they live in a food-limited environment, you know, requiring a lot of extra calories for either big muscle mass or a
Zoom
body
in
plan
there.
that would require a lot of active swimming to stay in the water column.
This one's up higher.
Are both
Go
pretty
get this one.
expensive. So. These are so cool. They look like gumdrops.
Oh, that's nice.
Nice.
Turn down the... He's really close. And getting closer. I'm widening
Great
out.
view.
He's right in front of us.
So question to clarify a little, Amanda, what you said. People are wondering, you know, these animals are pigmented the way that we are seeing them. It's just that in their normal environment, when there's not a big ROV booming a bunch of lights down here on the seafloor, there's not a lot of light reflection. So it's that they would not appear. So us carrying our own lights makes things look true, and we do a color balance with whites and darks so that they are true for our broadcast cameras, so that we are looking at true color based on a color temperature that is like sunlight at the surface. If we were down here with lower light or with a smaller light pool, lots of things would just look darker overall, not as much vibrant color available. viewers watching from all over the world here, which is super cool, including some of our partner sites. So hello to those in Santa Barbara. Scott Simon is on right now, representing UC Santa Barbara. Super cool. 2, 3.
Zoom in there.
Hmm.
What is that?
Oh, might have been a little eel pout. I don't know what this is. Mucus.
I
Mucus.
think this is a discarded larvation house, also called a sinker.
Yeah.
So larvations are swimming relatives of sea squirts, and they have this mold on their head
Oh,
made
sea
of
spider.
cells called
Oh,
oikoblast.
yeah.
Also, there's a sea spider.
Trade them out.
That might be more exciting than a mucus
Photobomb.
house.
That's all good.
All right.
See a spider.
Zoom in there,
cool
Wayne.
All right.
No, larvations are amazing.
They are amazing.
I like that.
But
This
these
is
are
pretty to look
But
at.
come on, picnic on it. See stuff.
Wow.
Very
So-
cool. Spiders.
Amanda has let us know that because their body is so small, some of their organs or gonads extend into their legs.
Yeah, and they're sometimes actually see-through. You can actually see the gonads, and you can see the gravid females. You can actually see the eggs packed down into the legs. My partner did a bunch of work on
Very
these
good.
type of
Okay,
animals in
full wide.
Antarctica.
Coming wide.
Have to go catch up.
Yep.
I'd like to bring our bearing around to 055. Is that okay?
Sure thing.
Sure.
All right. Bridge nav.
Wow.
There are folks in 28 countries watching Nautilus with us right now. So cool.
Wow.
There's an octopus there on the left. Several sea cucumbers on the seafloor here. The white ones look like they have oral arms that have feathery projections on them. So those oral arms capture food on them and bring the food to the mouth, sort of like licking your fingers clean after eating with your hands. They sort of do that with their oral arms.
Yeah, they can also clean off their legs and scrape along them because they diffuse oxygen through their exoskeleton there. And they can get biofouled. Their bodies can get biofouled, so they can use those little pinchers and scrape off their legs to sort of like taking a deep breath, clear off.
What's that, the pycnogonids?
Mm-hmm.
Ah, oh, neat.
Hello, octopus.
Oh, I haven't seen any of those lately.
They've been scattered about.
All right,
But
zoom in.
This one,
Okay, zooming in.
this one's a little curled up.
Yeah.
Turn down a little bit.
That's okay.
Uh-oh.
We're going to blow them out.
Maybe we'll see that third arm.
Now it's on the move.
Oh, run away. Oh, no.
I think
Okay.
I saw a little something.
All right.
All right, coming wide. Full
Oh.
wide. Full wide.
All right, thank you.
Excuse me.
And, yep, full wide.
Do you think it was different from the ones in the octopus garden, the moose octopus?
That looked similar.
That one looks similar to me.
Okay.
A good question about why the video can't just zoom at will. There are some ROV systems that have two different types of cameras on them. One is for zooms and pretty pictures, and one is for piloting. Ours serves a dual purpose, so video can't just zoom without the confirmation of the pilots because they're using that for reference, and particularly as they're approaching different animals or ledges, you know, they're using these different camera perspectives to line up. It's kind of like imagine driving your car with no control over your, I don't know, headlights or windshield wipers.
Go ahead, zoom in there.
So there's a lot of communication that you hear going on, and a lot of coordination between watch leaders
sea
setting
star
the
is not
priorities,
glitching.
what do we want to look at,
Looks a little worn.
and
Yeah.
the video engineers
Seen some battle.
controlling
Yes,
iris and
Who battles a sea
zoom and
star?
focus. Somebody
Yeah,
wants to lose.
That's
Interesting. Unless they can really
Okay.
coming wide.
pick up.
Full wide.
Science team in the back, a question coming in about how ubiquitous are these animals? Are we seeing mostly species that are unique to, you know, the Monterey Bay or California region, or are we seeing creatures that are the same across all of the seafloor?
Yeah, so far we don't have any species that are endemic to the seamount, meaning they're only found here. They are found in other parts of the deep sea off our coast and maybe the world, like some of these grenadier are worldwide. So yeah, nothing endemic, but we're always looking for something new.
But their densities can be much greater on the seamount, right?